Hello, everyone. My name is Li Lei. I'm bioinformatics scientist in Department of Allergy, Joint Genome Institute. Today, my talk is about genome-wide comparison of artificial induced and natural occurring mutations in Brachypodium distachyon. So when you shopping in the grocery see so many types of vegetables, cabbages, broccolis, cauliflowers, etc. What do you think? Are those belong to different species? The answer is no. All of them are bred from the same species, Brassica or nonsyn, by selecting the phenotypes caused by the natural occurring mutations. Modern breeding still relies heavily on genetic diversity that occurs spontaneously in nature. However, spontaneous mutation rates are extremely low, on the order of 10⁻⁹ per base pair per generation. Beyond that, natural selection against the variants that may be beneficial in the agricultural setting but deleterious in natural environment. Therefore, it's necessary to develop approaches to increase natural plants' genomic diversity and accelerate the development of improved cultivars. In 1927, Mueller found that the radiation caused the fruit fly have the red eye color. A year later, Steiner applied this radiation to treat the barley and maize, which started the new era of utilizing the induced mutation to breeding. Since then, induced mutation were widely spread, adopted as crop improvement tools. So what mutagens normally people use? There are three classic types of mutagens: two chemical mutagens, pseudomyce and EMS. These two cause the point mutation, and then the third one is the radiation. That is fast neutron. This usually causes big deletion. Those mutagens has been used for 200 species, and many of these mutant varieties have made enormous economic impact on agriculture and food production. Then the question is, are those induced mutations differ from natural occurring variants? If yes, how? In order to answer this question, we choose Brachypodium distachyon as the model system because it's small and have rapid generation time, deployed selfing, and a small genome, and efficiently transformed. So then we create the artificial induced mutant lines using the three classic mutagens. The pseudomyce that we create 1,833 lines, and EMS 48 lines, and fast neutron 39 lines, and then collect the seeds and grow them until the third generation, and extract the DNA from the third generation, and then do the Illumina whole genome sequencing. And finally, cross-snip with this genomic data. Beyond that, our group have already published the pangenome data from Brachypodium distachyon diverse lines, so we can make use of this data to compare the natural occurring mutations and then the induced de novo mutations. So here, we specifically ask us three questions. First of all, do induced de novo mutations have a preference on certain mutation type compared with natural occurring mutations? What are the functional impacts of the mutations? Do more induced de novo mutations tend to change phenotypes? And lastly, do different categories of pangenes have different functional impact mutations in both mutant and diverse lines? So to make the standing variants more directly comparable with the de novo variants, we split the standing variants into two categories based on the frequency. The first class is a common variance with some major allele count of at least 3 in our dataset. And this second class is real mutations with minor allele identified in either 1 or 2 accessions. The reason we split this is because the real variants have experienced a few generations of selections and are likely more similar in terms of mutation spectrum and context to induce mutations. So the first question we ask is that do de novo mutations have a preference on certain mutation type compared with standing mutations? In order to answer this question, we plotted the mutation spectrum for both de novo mutation and standing mutations. Since majority of the mutations are SNPs, so we only focus on plotting the mutation spectrum of the SNPs among different mutant and diverse lines. The y-axis is a fraction of the SNPs. We can see that for both de novo and standing mutations, transition have a way much higher proportion than transversions. And the majority of the mutations are concentrated in the C2T or G2A categories, which is consistent with the previous studies in sorghum, rice, soybean, and other crops. For de novo mutations like pseudomyces and the EMS, these tend to have preference on C2T or G2A mutation because pseudomyces and the EMS have way much higher proportion of C2T or G2A proportions than the standing mutations and then fast neutron induced de novo mutations. So for the first question, our answer is that yes, de novo mutation induced by pseudomyces and the EMS have a preference on C2T or G2A mutations. The second question, what are the functional impact of the mutations? Do more induced mutation tend to change phenotypes? So for mutations, as a biologist, we care more about the organism's phenotypes and if the mutation change the phenotype dramatically. For example, if a mutation causes a stop codon, premature stop codon starting lost, or frame shift or gene fusion, this definitely will cause the phenotype change. However, for non-synonymous mutation, we know they definitely will change the amino acid, but we're not sure if those amino acid change really cause the phenotype changes. So in our project, there are huge number of the mutations that are non-synonymous, and we need to figure out which non-synonymous mutations tend to change the phenotype. So in order to annotate the mutations, we firstly run SNP-if to predict which SNPs cause the frame shift to stop gain or loss, starting loss, the non-synonymous SNPs, etc., and etc. And then we run another tool called as bad mutation we developed before to see if non-synonymous tend to change the phenotype. That means they are deleterious. So the rationale for the bad mutation pipeline is that assume the deleterious mutation tend to change the phenotype, and at the same time, they tend to disrupt the genetic conserved sites. If you want to know more details, please check our paper and also the GitHub repository for this pipeline. So based on the annotation by SNP-if and prediction of the bad mutation, we plotted the proportion of the different type of the mutations. The left panel shows the proportion of mutation annotated by SNP-if. The left side is a SNP, and then the left side is for SNP, while the right side is small deletions. X-axis is a proportion of different type of the variants. Y-axis is different categories of the mutation. The purple color indicates the high impact mutations, and we can see that a very tiny proportion of the variants are high impact mutations. That is annotated by SNP-if, and for around 15% of the SNPs are non-synonymous, and we're not sure how many of them are deleterious. So then we run bad mutation pipeline. This is the right panel. I show you how many of the non-synonymous SNPs are deleterious, we can see that at least 21% of the induced mutations are deleterious, and this is much higher than the standing variations, which makes sense since those are artificial induced mutations. And this answers the second question we asked: do more induced mutations tend to change the phenotype? Yes, it does. And last question is that do different categories of pangenes have different functional impact mutations in both mutant and diverse lines? To answer this question, we follow the way our group did for categorizing the pangene based on the frequency. That is, if the pangene shows in all accessions, that is a core gene. And then if the pangene shows in 53 and 55 accessions, then we define it as a software core gene. If showing in 3 to 15.2, that is a cloud, that is a share gene. And then if just showing in 1 or 2 accessions, then defined as a cloud gene. And based on the classification, we hypothesized that a core gene cluster tend to be most conserved, have most phylogenetic conserved site, and have lower deleterious load, and the cloud gene are more lineage specific and have least phylogenetic conserved site and higher deleterious load. So to test this hypothesis, first we need to define the conserved site. So for each gene, if we do alignment for the 19-1 available angiosperm species genome for each site, if they have the same nucleotide at least 21 species, then we define this site as a conserved site. Let's like here. This is consistent with bad mutation criteria. If the gene contains at least 1 conserved site, we define this gene as a gene with conserved site. So for gene with conserved site, we compare how many of the conserved site per base pair they do have. We can see that the number of the conserved site per base pair decreasing along the core, soft core, share, and cloud genes. So accept the hypothesis that core gene tend to be more conserved, while cloud gene tend to be more lineage specific. And then we will answer the question we proposed before. Do different categories of pangene have different functional impact mutation in both mutant and diverse lines? Firstly, we will see the diverse lines. We plotted the deleterious SNPs normalized by conserved site per base pairs. We can see the cloud gene tend to have most deleterious SNPs, while core gene have least deleterious SNPs. That makes sense, suggesting that purified selection working on differently according to the conservativity of the gene. Core gene tend to be more conserved and they experience more stringent purified selection, and so most of the deleterious SNPs could be purged, but opposite to the cloud genes. To the mutant line, we will see there is no significant difference of the deleterious SNPs for each categories of the genes of the pangenes due to lack of the natural selection. Okay, let's summarize the results. So we identified 2 million mutations from 2,000 mutant lines, and we predicted the functional impact of the huge number of non-synonymous SNPs. And this will increase the utility of the mutant collections. And also C2 and the EMS tend to induce C2T or G2A mutation. Induced mutation tend to be more randomly compared with natural occurring mutations. And in the future, we will collect structural variants in fast neutron lines and look for the context motif surrounding the mutations. We will check the phenotype of mutations in certain important genes. With that, I would like to thank all of the folks mentioned here, and also I'm happy to take any questions. Thank you.